So, welcome to the new session of the cell, the unit of life. And today we'll start from the mitochondria. Yesterday we have ended into lysosome, and so today we'll start from mitochondria. Okay. So, mitochondria. What do you mean by the mitochondria? Mitochondria is a part of the cell organelle that is responsible to provide the energy to the cell. So, mitochondria it is present in eukaryotic cells only. Eukaryotic cells only. In prokaryotes, I have made you study in the lecture 1 or lecture 2 that in prokaryotes the function of the mitochondria or the power of the cell is generated in the mesosome. So the mesosome structure is actually present in prokaryotes, and the eukaryotes only have mitochondria. In prokaryotes, the function of mitochondria is carried by mesosome. Okay, the mitochondria I will write in the short form as M. Okay, not visible under microscope if not stained. So if we do not stain mitochondria, then it is not visible even under the microscope also. Okay, to visualize the mitochondria, we need to stain it with dye. ATP is synthesized in mitochondria. ATP is synthesized. It is also called as the powerhouse of the cell. Powerhouse of the cell. Okay. So when we require more power, see in the real life, we require more power when we do extensive work, or when we work vigorously, we need more power. When we run, when we swim, when we do more difficult work, we need more energy. So from where the more energy is being generated in our cell? The more energy is being generated in our cell from the mitochondria, and the mitochondria, the energy is being generated from carbs, that is carbohydrate, proteins, into ATP. I will make you study those Krebs cycle and the other energy produced, the EMP cycle, that is the glycolysis, in the respiration chapter. But for now, you study that the ATP is being generated from the carbohydrates, the food we take, the protein we take, the lipids we take, the foods, the ATPs are being generated from these only. Okay. The structure of mitochondria. The structure of mitochondria. It is cylindrical or sausage like structure. Okay, it is cylindrical or sausage-like structure, but its shape vary in different सेल. जिस सेल का जितना जैसा काम करता है, जो सेल जो काम करता है उसके माइटोकॉन्ड्रिया वैसे ही दिखता है. जो सेल ज़्यादा काम करता है उसमें माइटोकॉन्ड्रिया भी ज़्यादा है. जो सेल कम काम करता है तो उसका माइटोकॉन्ड्रिया भी कम होता है. ठीक है, अभी स्ट्रक्चर में आइए. सो लेट अस ड्रॉ द स्ट्रक्चर. Mitochondria is a double membrane structure. So if we draw mitochondria, okay, this is the outer membrane. This is the inner membrane. So the inner membrane has some foldings. Okay, the inner membrane has some foldings. These foldings are invaginated towards the inside of the inner membrane, towards the matrix level. So inside the inner membrane we call it as matrix, and outside the inner membrane we call it as peri-mitochondrial spaces. Okay, so this is the matrix. And these spaces, it is peri-mitochondrial spaces. Okay, so here we have ribosomes also. Yes, here we have ribosomes also. A ribosome, and it is 70S. In mitochondria we have 70S ribosomes. Okay, these are the cristae. So what is the function of the cristae? Cristae are the foldings which is inward to the matrix, and these foldings actually increases the surface area of the mitochondria. So mitochondria is a structure, is small structure. These are being the surface area of a small structure is being increased by these cristae. Okay, these are called the infoldings. Okay, so it's a double layer membrane. What to remember, it's a double layer membrane. It is present on the eukaryotic cells. It generates the ATP. It has some infoldings called the cristae. Cristae actually increases the surface area of the mitochondria. Okay, so now you can differentiate. See, matrix is the inner compartment. Matrix is the inner compartment, and peri-mitochondrial space is the outer compartment. Okay, these are being filled with aqueous fluids. Okay, so now we differentiate between difference between outer membrane and the inner membrane. Outer membrane and the inner membrane. Okay, so it has a delimiting layer, absent in inner membrane. Okay, porine proteins are present in outer membrane, absent in inner membrane. Due to the porine protein, there we can see some facilitated diffusion. Okay, facilitated transports are being seen due to this porine proteins. Okay, permeable, more continuous, less or discontinuous. IM, invaginate to form cristae, increases the surface area. Surface area is increased. Okay, compartment is formed due to inner membrane. Multiple compartments are being formed due to the inner membrane. Compartments are formed due to inner membrane. Okay, so now we come, outer membrane and inner membrane had different type of enzymes. Make it a note. Outer membrane and inner membrane has different type of enzymes. Okay, now we come to matrix. So what is the matrix? Matrix at the innermost position of the mitochondria. It lies to innermost side of the inner membrane. Okay, homogeneous. It is homogeneous substance. Inner part. Single DS circular DNA now converts to mRNA 70S protein. It is semi-autonomous organelle. mRNA, उसके बाद आते हैं rRNA, tRNA and few RNA. Okay, it is semi-autonomous organelle also. It is also a semi-autonomous organelle. So matrix is a semi-autonomous organelle. Okay, now it is also, you can say, as self-duplicating organelle. Self-duplicating means it divides by fission. Divides by fission. Okay. Now I am drawing a structure. This structure, see, the structure of the mitochondria. Okay. Now These are heads. These are the heads. These are the stalk. Okay, so the matrical head of the matrix. Okay, let me draw it good. I'm good at drawing, but not too much accustomed with this technology, writing in tab. Okay, this is the head. This is the head oblique F1 subunit. This is called the stalk, and this is the base F0 subunit. Step: ATP synthesis. That is the oxidative photophosphorylation occurs here. Phosphorylation occurs here. Okay, it is a part. It is a part of electron transport chain, etc. Site is the inner membrane. Site, inner membrane. Inner membrane. Okay. Now F1 oblique F0 particle F1 oblique F0 particle oblique oxysome ATP synthetase. Enzyme is being secreted by the F1 and F0 particle. Okay. The matrix, write a note. The Krebs cycle. The Krebs cycle occurs in the matrix of the mitochondria. Okay, now we come to ribosome. Now we come to ribosome. 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 Ribosome, it's a membraneless particle. It is a membraneless particle. Ribosome do not have any membrane. Protein synthesis occurs here. Protein synthesis occurs here. Okay, it is smallest. Who discovered it? Discovered by George Pallade. George Pallade in 1953. Okay, is a densely stained structure. It is granular. Okay. Now S is called the Seedberg unit. Seedberg unit or sedimentation unit. Now in prokaryotes and in eukaryotes. In prokaryotes it is 70S. In eukaryotes it is 80s. Okay, now it is 30s divided into 30s and 50s. It is divided into 40s and 60s. Okay, this is a smaller, this is larger, this is smaller, this is larger. Okay, this is rRNA. It is 16s, 5s, 23s. In prokaryotes, in eukaryotes, it is 18s, 5s, 5.8s, and 28s. Okay, remember these things. These are being asked in the NEET. Very much important. In proteins, protein, 21 protein, 34 protein. Here 33 protein and 47 protein. This is ATP. Okay. Now note, rRNA plus protein both forms ribonucleoprotein. rRNA and the protein, it is ribonucleoprotein. Okay. rRNA and protein is ribonucleoprotein. Okay, got it. Now we come to vacuole. 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 It is also called as sap vacuole. Sap vacuole in plants. Membrane bound. It is membrane bound. Membrane bound space in cytoplasm. Occupy 90% volume in plant cell. It is single membrane bound. The next point, H2O, sap, inorganic ions, waste material, excretory material, which are not useful for cell, are being deposited here. Okay, it's a cytoplasm, bracketed low, and a vacuole high. Against movement, against concentration gradient, that is ATP is required. Uphill transport, it is also called as active transport. Okay, it is also called as active transport. Now we come to contractile vacuole. Here is a heading as contractile vacuole. Contractile vacuole. Okay, there is contractile vacuole. This is the cilia. Here H2O pair. Example, amoeba, paramecium. It is the mouth. Here it is mouth. Okay, here it is mouth. So what it becomes? Excess water absorb and pump outside the body. It is osmoregulation. Release waste material. That is excretion. Now we come to food vacuole. Give it a heading as food vacuole. Food vacuole. So what is food vacuole? This one is food. Okay. Now food comes here. It will engulf it. It will just engulf it. This is the plasma membrane. Invagination of plasma membrane. Invagination of plasma membrane. This structure is separated from the membrane. This structure is separated from the membrane. Now this one is lysosome. This is food vacuole. Okay, engulfing. It is actually I have made here is engulfing of the food material. The food material is being engulfed here. Engulfing of the food material. For example, you can give it as protista. So today we have studied about the mitochondria as well as the ribosome, and on the later part we have ended it in the vacuole. Okay, in the next lecture I will continue from the later part, and in the end I will make you revise the whole chapter in a gist, in a simple video, in a small video of 30 minutes. I will made all the gist in that 30 minutes, so you can memorize and take it and take the notes for the last minute preparation before your exams. Okay, so wish you all the best and hope you will all see tomorrow. Thank you.